This is a very different presentation than one I usually give because I usually focus much more on the therapeutic side, but I've tried to sort of start out at least with the core technology and also highlight aspects that I think are relevant to the synthetic biology audience. So let's start at the beginning. We sort of think about the genome as a scaffold or a chassis that nature has already generated for us that we could make very specific changes to create a biological system that we're interested in. And that might be a system that the synthetic biology crowd want to make some specific change to. The way we think about that is this is a drug target. There are monogenic diseases caused by individual changes in the genome, and we need a technology that can move to that particular location and exert a change to the genome at one site, despite the complexity and the size of a mammalian genome. And so the technology that we've been focusing on is a natural one. It's zinc finger proteins. These are a natural class of transcription factor. They're the most abundant type of DNA binding domain found in the human genome. They, in fact, outnumber all the other types of DNA binding domain put together. And so what you're seeing here is nature's ability to make use of this domain essentially over and over again to create new transcription factors that operate and bind to different sequences in the genome and carry out different functions. And it's this natural versatility that sort of lends this domain to engineering principles. So Carl Paabo, when he was here actually almost 2 decades ago now, solved the crystal structure of ZIF268, which is the first 3-finger C2H2 class zinc finger protein, solved when bound to its target DNA sequence, and that's what's shown in this slide here. This is the ribbon representation. The zinc finger is shown in green, and the 3 fingers are shown off to the left behind the DNA, which is shown in blue. And I'm focusing on finger 3, which actually is the bottom right-hand corner. So each finger actually is a very compact domain. We think about this as having a very consistent structure, and it's essentially its job is to present the same face of this DNA binding helix into the major groove of DNA. And in fact, the yellow region I've highlighted at the end of that finger is the section which generates the sequence-specific DNA binding function. And if you make mutations to the amino acid finger in that region in yellow, you can create a different zinc finger protein that recognizes a different 3 base pair step. A second feature, which is not so obvious from the structure, but is very critical to the engineering of this domain, is that it's highly modular. So each finger essentially is a separate DNA binding unit that recognizes 3 to 4 base pairs worth of DNA. And so there's no requirement for symmetry in the DNA binding subsite. And so that allows us to essentially produce tandem arrays of these proteins and expect them to recognize a sequence, which essentially is the sum of the parts. And so that's really what's shown on this slide. That on the left-hand side, I'm tilting the DNA, tilting the zinc finger up to show the critical amino acid positions of -1, 2, 3, and 6. If you make mutations in those positions, those create a different array of amino acid side chains which interact directly with the basis of DNA. And that gives rise to this concept of a design or a finger design in which, depending on the precise amino acid composition of those critical positions, that determines the triplet site specificity. Nature's, of course, never plays that easily, and in fact, in this particular situation, that's true also. And so, in fact, to make life easier, we actually use a 2-finger module array to build proteins. By having the 2 fingers next to each other, it allows us to ensure that each of those 2-finger modules has been pre-characterized to play nicely with each other, so that there's no conflicting interactions between any of the 2 fingers. But in principle, we just have a large archive of these proteins, these 2-finger modules, and we can string them together to create long DNA binding domains that recognize extended DNA footprints within the genome. And so in the example I'm showing you here, this is an 18 base pair binding protein. And if I avoid repetitive DNA sequences in the genome, that's sufficient to specify a unique site. So what this allows you to do, in principle, is to create an addressing activity, a way of loading a particular protein to a defined site in the genome. And then the next question is what happens when that protein gets there. And that's really determined by a second feature of transcription factors and DNA binding domains, and that is that they're highly modular. So I can take the DNA binding domain, the zinc finger domain, and I can add to it different domains, be them transcriptional regulatory domains, so activators or suppressors of transcription, or I can add a nuclease domain and actually cut the genome at a specific site determined by the DNA binding specificity of the zinc finger itself. And so for the first half of this talk, I'm actually going to focus on the transcription factor side and take an example of gene repression where the target actually is a monogenic disease. It's Huntington's disease. So this was a seminal paper in the HD space which demonstrated that if you overexpress the mutant form of the Huntington gene, then that is sufficient to drive the sort of phenotypic consequences of Huntington's disease in the mouse. And one of the phenotypes they have is this clasping defect. But perhaps more importantly, what this paper also demonstrated with this inducible system is if you shut off the activation of this, or shut off the expression of the mutant Huntington, not only does the animal, does the disease halt, in fact, the animal actually recovers a substantial function. And so this really has been the pioneering sort of result that's led to a drive to find mutant Huntington lowering strategies. So can we shut off the expression of this mutant gene? So that's actually more complicated than it sounds. Whilst Huntington's a monogenic disease, it's caused by a CAG expansion in exon 1 of the Huntington protein. And unfortunately, the wild-type Huntington gene also has a CAG expansion. So it's not simply a challenge of shutting off the mutant version. One has to discriminate between the wild-type version, which is can have up to about 28 repeats without being pathogenic, or about more than 40 repeats, which is when the protein becomes pathogenic. So one could intervene at many stages, and there are very, very many therapeutic approaches aimed at either addressing the mutant protein or the RNA encoding that protein. Obviously, as I've described, we focus on the DNA as being the target. And in fact, the idea is to use a transcriptional regulator to shut off the expression of the mutant allele in a selective fashion. And so this is what we're going to try and do. The disease allele obviously has this much larger CAG expansion, and we'd like to try and target that. And we're going to actually target the CAG repeat itself with a zinc finger protein that targets CAG, and then ask that protein to discriminate between the length of the CAG repeat. In other words, have it be, if you will, a yardstick for how long that CAG repeat region is. Just to show you how we can measure this, we in fact have, thanks to a big effort that the NINDS and CHDI initiated, there's a large number of fibroblasts and associated iPS cells from HD patients that have a vast array of different repeat lengths of both the mutant allele and wild-type allele. And we can make use of the occasional SNPs that occur in the wild-type and mutant alleles to identify TACMAN primers that allow us to distinguish specifically between the mutant and wild-type forms. And so in all the experiments that follow, this is going to be the way it looks. I can measure in gray the total Huntington output, so this is just how much of Huntington's in the cell. But I can also, using these individual primer probe sets, measure how much of the mutant or the wild type is. And mutant's going to be in red, and the wild type in blue. So in this experiment, what I'm showing you is that we have generated an allele-specific repressor that leaves the wild-type expression alone, but substantially over 90% suppresses the mutant. And I've made a biallelic suppressor which actually knocks out the expression of both. And that can also be observed at the protein level. This is a Western blot showing that the mutant levels are lost in the allele-specific, but both mutant and wild-type are lost with the biallelic. So this is showing experiments done in a line that has a CAG 15 on its wild-type allele, and a CAG 70 on the mutant allele. But in fact, the human population is much more broadly spread than that. The wild-type alleles are anywhere from about 10 to 28 or so, and mutant starts at around 40. And so what we'd really like to do is find proteins that can discriminate across a much broader range. 70, of course, would be way out to the right in this particular distribution. So we've collected these lines from Coriell and done the experiment where we introduce the zinc finger proteins to these lines and ask whether they continue to show discrimination across these different repeat ranges. And the answer is that they do. So this is just showing you again at repeat lengths in the mutant allele range of 70 or 67 repeats. Everything's working just fine. the red mutant alleles being repressed, the wild-type alleles being spared. But even when we get out to repeat lengths of 40 or 45, we can continue to show this discriminatory behavior. So this is looking very encouraging for the way that these proteins are interacting, at least as in the way that they discriminate between the wild-type and the mutant alleles. Of course, CAG repeats are not unique to the Huntington locus, and so it's also necessary to be discriminatory not just within Huntington, but also across the whole genome. And so we did a bioinformatics search looking for the most, just blasting the exon 1 sequence of Huntington's CAG repeat against the genome, and asked for other CAG repeat-containing genes that are expressed in fibroblasts that had a CAG repeat within about 10,000 base pairs of a promoter. And so that was this list here, and I'm showing you both the CAG repeat length at the bottom and the position relative to the start site. And the good news here is that with the exception of one gene, this SDC1 gene, we see very good discrimination against all these other sites as well. So these proteins that are not repressing the wild-type Huntington allele but are repressing the mutant also show good discrimination genome-wide. This, of course, is the sort of looking under the lamppost approach. We've selected genes based on a bioinformatics search. You can also do Affymetrix-based array technology. And I'll show you 3 examples here. In the middle is a protein that we have that has very high affinity and demonstrates a much broader specificity in the sense that it regulates other genes. We have 2 other variants, one to the left and the right, that regulate 3 or 2 genes in the entire genome respectively. So it's possible to create these proteins that recognize the Huntington CAG repeat, discriminate against the wild-type repeat, don't touch the other CAG repeat-containing genes in the genome, and leave the other genes in the genome essentially untouched. And this is just drilling this down to, obviously what I'm showing you with the Affy experiments is one concentration at one dose. I can also do these experiments over a much broader dose range. This is using 5-point dose titrations, demonstrating that for the 30640 protein, which is the one to the far left, we can see very good discrimination at the top between wild-type and mutant Huntington. And you can see that the gray, the THAP1 protein, is not touched at all, but SDC1 is. But if you move across to 33074, which is the middle protein, we don't see any suppression of that potential off-target. Okay, so do these proteins work in vivo? It's possible to test that using the mouse models. Just a quick note on mouse models in Huntington's disease. It turns out that human-length CAG repeat expansions cause no disease in mice. And so, in fact, the disease-causing mutations are much longer. And so for the experiments I'm going to show you, we'll be testing 2 things. The R62 model, which is the classical model of Huntington's disease. It has a 120 CAG repeat expansion. But to test specificity, to show these proteins continue to discriminate against wild-type arrays, we're going to test also this HDQ20 model, which is a 20 CAG knock-in into the mouse allele. So I'm going to look at 2 proteins. These represent 2 different types of protein in terms of their dose responsiveness. In the experiments that are shown, they're either called ZFP1 or Class 1 or Class 2. ZFP1 is actually 30640. It shows you a very broad dose range, a very broad discrimination, and is most active between about 3,000 nanograms and 10 nanograms. And then we have a second, which is this 30648. This was this very high affinity protein that showed less good specificity in the Affy experiments, but operates at very low levels, so between 3 nanograms and 0.1 nanograms. So we just checked first to make sure these proteins actually worked in neuronal cells in the dish. So these are R62 neurons taken out and grown in culture, introducing the ZFPs and showing that both classes are capable of repressing mutant Huntington. So this is the animal studies. We're now using a bilateral infusion of AAV6 containing the zinc finger proteins and encoding these particular repressors of Huntington's disease. And on the left-hand side, you're seeing the repression of R62, showing about 50% or so suppression for both the 640 and 648, whilst compared to GFP, whilst leaving the mouse Huntington alone. So that's only about a CAG7 repeat. In addition, we can switch model and go into this knock-in model, which is called the Q20. It's actually a CAG18. And in this setting, once again, on the right-hand side, you can see that the mouse Huntington allele is not modified. And the 640 shows very good discrimination against this Q20, showing only about 20% suppression in this particular setting. So we're starting to show proteins that can actively suppress the mutant Huntington, but also leave the wild-type Huntington lengths alone. So on the ZFP-TF side, what I hope I've shown you so far is that we have the ability to downregulate, or we could also do upregulation of any particular target gene of choice. It's possible to get remarkable selectivity, both in vitro and in vivo, using these proteins. They're tunable by modulating the affinity and the specificity of these proteins. And obviously, these are potential tools for the development of synthetic regulatory circuits, as actually Pam described. So in the latter half of the talk, what I'm going to do is talk to you now about work on the left-hand side of this slide, which is the zinc finger nuclease front. So it's exactly the same core technology, so the ability to build a zinc finger DNA binding domain that specifies a particular site in the human genome, but this time attaching a nuclease domain which will produce a targeted double-strand break. And we can use that break to essentially achieve three outcomes: the knockout of a gene of interest, the correction of a mutated gene, so just changing a few base pairs, or the insertion of an entire gene or potentially a series of genes to a defined location in the human genome. So zinc finger nucleases are actually a dimer of proteins that we have to build. That's what's shown in the top left-hand side here. That to create an active enzyme, we actually have to force the dimerization of these two enzymatic domains, the FOC1 domains. And to do that, the proteins have to bind DNA in the correct orientation and spacing. And that's a very constrained situation. You need a spacing of about 5 or 6 base pairs to achieve appropriate dimerization. A 7 base pair spacer, for example, shows no cleavage activity in most assays. So that gives us the ability to be rather specific, because it turns out that the binding of the protein on the left, for example, by itself, elsewhere in the genome, actually isn't even sufficient to nick DNA. The dimerization event is necessary for the enzyme to engage DNA in a cleavage mode. And so we can use this cut, and that's what's shown at the top here. This is taken from a review written by Dana Carroll, who's one of the pioneers of using double-strand breaks to achieve these different types of genome editing outcome. And on the left-hand side, you can see that if you just let the nucleases do their job and let the cell repair that double-strand break, it does so using a process called non-homologous end joining, or NHEJ. This is simply the two ends of the broken DNA get glued back together again. And when that happens, there's no error checking to make sure there's not been information gained or lost at the site of the break. And so this is an inherently mutagenic process and can drive to gene disruption if you target that double-strand break to a region, a coding region in a gene, for example. Alternatively, you can take that same zinc finger-induced double-strand break, but in addition, add the donor DNA sequence, so a homologous donor DNA, that can be used in the process akin to homologous recombination, in which the information from the donor is essentially copy-paste into the genome at the site specified both by the nuclease and by the homology sequences in the donor. And so what I'm going to start with is a couple of examples of this homology-driven process. And if I have some time at the end, I'll talk about the gene disruption process. So the reason this process works is, in fact, that the molecular basis of homology-directed repair is, in fact, blind to the insertion of information between the homology arms. And so it doesn't really matter whether this is an X-ray-induced double-strand break or a zinc finger-induced double-strand break, or indeed any other tool or technology for introducing a site-specific double-strand break, TALEN, CRISPR, whichever your favorite method is. When you provide that donor DNA with homology arms, the only thing that the double-strand break does is cause the cell to search for those homologous regions. But the region in between those homologous arms is essentially blind to the cell, and the cell will faithfully copy whatever information you've included in the donor into the genome, believing it has found the sister chromatid, and believing that this is how to produce a perfect repair. And so that allows us to, as the investigator, to include information in the donor DNA that we wish to literally copy-paste into the genome at the site specified by both the nuclease cleavage site, but also by the homology contained in the donor arms. So this is an experiment shown in iPS and ES cells that was actually done locally by Dirk Hockemeyer when he was here in Rudolf Jenisch's group at the OCT4 locus. And this just I show you this because the efficiency of this process occurs is rather remarkable. You can see, obviously, there's one in the southern below. You can see one band that's of the inappropriate height. That's actually the only one that's inappropriately targeted. Everything else is exactly what we want it to be. And so this can go in with very high efficiency, and it allows you to do things like tag endogenous genes in different ways. So depending on how you design the donor DNA, it's possible to introduce reporters of OCT4 expression. So on the left-hand side, so I beg your pardon, on the right-hand side, you can see we've introduced an OCT4 2A GFP fusion. And so if OCT4 is expressed, the cell just glows green. But on the left-hand side, sorry, if you fuse OCT4 directly to GFP, you have actually a readout of not only the expression of OCT4, but where it is in the cell. And so it's possible using the system to tag endogenous genes in interesting ways to have a very precise readouts of biological function. You can also take this approach to sort of the quote-unquote the nth degree. This is an example of trying to eliminate a mutation found in human disease. It's the Z mutation in alpha-1 antitrypsin deficiency. In this particular case, we used a double-strand break to introduce, to stimulate the process of homologous recombination. And we also selected for those cells using a pure cassette and then removed that cassette using piggyback-based transposon excision. So the important thing here is that if you do that, you eliminate the functional consequences of the Z mutation, so you no longer get the accumulation of the polymeric alpha-1 antitrypsin. So that's what's shown what will be shown in the green in the central panel. And that those cells, when they engraft in mice, we can make them into hepatocyte-like cells, engraft them in mice, and they produce human albumin. I bring this example up because obviously one of the key features of any editing technology that produces a double-strand break is the specificity of that reaction. And in this particular case, we performed full exome sequencing of the edited cells and could find no ZFN-induced mutations outside of the intended mutation precisely at the Z mutation. And so this is just an indication this is the hardest we've looked for off-target sites thus far using this exome sequencing, at least in mammalian cells. So I've shown you so far examples in which we have either introduced a gene size fragment, something like a GFP, or we've changed a few base pairs, as in the case of the alpha-1 antitrypsin mutation. But what about entire chromosomes? And so in this experiment, we're thinking about Down syndrome. This is a trisomy of chromosome 21. And the thought was, well, is there something that we could do with an editing approach to actually bring about the elimination of an entire expression of an entire chromosome? And actually, there's a lesson from nature here, which is that in fact we know how to do this. The genome does it already, and that actually is exist-based gene silencing, in which one of the X chromosomes is completely silenced using this exist-based process. And so what we decided to do was to edit the chromosome 21 in cells from a Down syndrome subject, where we'd introduced into the extra chromosome 21 an inducible transgene encoding exist. So this was the experimental plan, that we'd take these fibroblasts, we'd add, using a zinc finger, the exist transgene to chromosome 21. This is a DOX-inducible cassette, so in the presence of DOX, we express exist, and we ask whether we can silence that extra chromosome. So this is all work done with Jean Lawrence at UMass. So if you're going to introduce a large transgene, this is a single-step integration of a 19 kilobase fragment that contains both the regulatable sequences and the exist cassette. Better confirm that it's gone in appropriately, as we did that by southern blotting here. And in all the experiments I'm going to show you in the next 2 slides, I'm showing you experiments in which we selected a clone in which just one of the 3 chromosome 21s contains its entire exist cassette. So, first things first, this is what you'd expect. This is a normal female cell demonstrating exist painting of the extra X chromosome. That painting forms sort of a hole. If you do staining for a protein called COT1, this is a heteronuclear RNA staining pattern which associates with active chromatin, and it forms this highly condensed section which is known as a bar body. This is the sort of the condensed chromatin form that you expect an exist silence chromosome to take. In our clones, we get exactly the same pattern. So this now is a male cell, so there is index inactivation of a chromosome, of the X chromosome. Now what we're seeing here is expression of X cyst only on this chromosome 21. And you can see exactly the same pattern. So on the left-hand side, fish showing that there's this zone of X cyst expression in cells with DOX that produces this similar hole and a similar condensation pattern or a bar body. And if you do further staining, so this is now for markers of repressive chromatin markers, all those markers line up and co-localize with the expression of X cyst. So the question then is, okay, so what I think I've shown you is that we've introduced this X cyst, so does it actually work? Do we see suppression of the chromosome 21? And the answer is we do. Now don't forget, these are cells that have 3 chromosome 21s. What we're comparing here is the cell plus and minus DOX. So it either is expressing the X cyst and is shutting down one of the 3 chromosome 21s or not. And so we expect about a third reduction in the expression. That's kind of what we see here. So if you can see that chromosome 21 is the only chromosome that shows this marked suppression when we remove the when we add X cyst to these cells. So what I hope I've shown you in the targeted gene addition space is that we have the ability to add information to the genome, and we can do that in a very site-specific way, which is capable of changing a single base, as in the example of alpha-1 antitrypsin, or we can add up to 10,000 bases to a given site. We can achieve single locus specificity that was shown by the southern blotting. And I think these are very powerful tools for the synthetic biology community. The genome itself can be manipulated, and thus becomes the chassis for future endeavors. And the applications can include reporter genes, safe harbor loci for the addition of genes to defined locations, and allows us to model genetic diseases and maybe even cure genetic diseases if we can achieve this in a therapeutic context. So in the last minute or so, I'm just going to touch very briefly on our work in HIV. We see the nuclease platform as a way of being a therapeutic platform, and our focus has been working in the HIV space. The technology that underpins this is exactly the same technology we've been exploiting across genome applications in many different organisms. It's the knockout of a specific gene, and this is a now highly sort of aging list of organisms that have been modified by this approach. So with HIV, the question that we've asked ourselves is the following. We know that there are great antiretroviral medications out there that are very potent at suppressing viral replication, but they don't actually cure the patient. The HIV remains present, and if you remove antiretroviral medications, the virus comes back. And so the question is, can we effectively, quote unquote, cure a patient by reestablishing an immune system which is HIV resistant and would allow the patient to mount a successful immune response to virus, and so rendering HIV to the same immune control that allows everyone in this room to suppress every other viral infection that you get. And so we have work in the clinic focused on T cells, which are, of course, the principal cell lost in HIV. But we also have efforts in hematopoietic cells as well, and just focused on that today. So the goal here is to take a lesson from nature, and that is that the CCR5 protein, which is a co-receptor for HIV entry, is known to be necessary for entry of HIV-1 into cells. And there's a natural mutation called CCR5 delta 32, and this mutation is present in the Caucasian population at about 10%. About 1% or so are biallelic or homozygous for this mutation and resist HIV infection. And so the idea is then that we take this HIV-resistant, these resistant cells and put them back into a patient that has HIV and allow them to become HIV negative, and that experiment's been done. There's a study done in Berlin that demonstrated that this transplant of stem cells from a patient who had this CCR5 delta 32 homozygous state was sufficient to eliminate HIV in that patient. The patient's now 5 years out and has no detectable HIV. And so our idea is to try and sort of reproduce that. And so what we're doing is making nucleases which cut the CCR5 gene, allowing this NHEG process to repair the gene and disrupt it, and in this case, essentially phenocopy, knock out the CCR5 location. And the idea then is that we can do that in hematopoietic stem cells, and by doing so, provide the patient with the ability to control HIV. So this is a collaboration between Sangamo, Paula Cannon's group at USC, and John Zaya and Dave DeGusso's group at City of Hope. This is a CIRM-funded disease team. And I'll show you one experiment in which we've taken human cells, put them into the NSG mouse model. If you just put them in with HIV and you don't do any modification, then what happens is HIV takes off. You see lots of HIV in the bloodstream as the HIV infects these human cells in this NSG mouse model. You do the same experiment, but you first modify the cells with the nucleases so that they no longer express CCR5. You get the following pattern, which is shown in red, that after about 6 weeks of the virus taking off, it crashes, and in fact, by 12 weeks, we can no longer find HIV in the bloodstream, and we can't find HIV in the tissues of the animal either. As you'd expect for this sort of selection, that during the expansion of HIV and ultimately as the CCR5 negative cells sort of win this battle, they are selected for. And so, as you can see at the bottom, with those very dark bands, this is an assay looking for disruption of the CCR5 locus, and you can see that the selection has taken place very potently. This is all published in Nature Biotech a couple of years ago. So we have 2 studies underway. We have some clinical work that will be actually presented next week at the ASGCT meeting, and the CD4 T cell work. So that's the same idea, but just modifying the T cell population in these patients and ex vivo and putting those back. But we also have this AIDS lymphoma effort with CIRM, which is on its way to the clinic, hopefully next year. So what I hope I've shown you is that you can take an editing technology, and there are numerous technologies out there now. You can use that core DNA binding activity to create transcription factors that can obviously regulate gene expression in an investigator-driven manner. But you can also create nucleases that allow us to consider editing the genome itself, so editing this sort of natural chassis that we have for synthetic biology applications. So many people to thank. I'll just focus on a couple. All the Huntington's work was supported by CHDI and my collaborator there, Ignacio Munez. I've obviously mentioned work from the Wistar and MIT with Rudolf Janisch, and with Jean Lawrence at UMass Medical Center. And I didn't talk about the clinical work today, but I'll pause now and answer any questions.
Can you comment on which one will work better? Like, not will work better, like which one you think can work better, either CD4 knockout of the CCR5 or the stem cell version?
Well, that's a great question. So I think that the stem cell approach has the most clinical validation because of the work done in Berlin. And so, although it is, of course, an N of 1, right? So we have one patient that's been cured using this stem cell transplant. The T cell work, relies on the idea that obviously the major cell lost in HIV are CD4 T cells, and the major functional loss is the lack of help for CD8 cells that are HIV specific. I think that approach should work nicely in cells as long as the immunological status of the patient is not too severe, it's already severely compromised. I think we're going to need a suitable, quote unquote, protoplasm to put those cells into to give the patient the ability to control HIV. So I think that they're quite different. Very hard to tell.
Terrific talk, Phil. I had a question. You're already obviously through your IND and into the clinic for the CCR5 knockout. I just had a kind of a curiosity. When you described the technology to the FDA, were there any questions about variability in the lesion CCR5 site from cell to cell due to changes in NHEG mechanism or indel introduction?
Yes, absolutely. So, raising a very good point, that the NHEJ-based approach is imperfect or imprecise in exactly what modification it makes on a per cell basis. And so what we did was we characterized that mutation for thousands of alleles, And as you can imagine, for the vast majority, there's a set of potential outcomes that each, there's even large deletions. Most short deletions give us very defined truncation points based on when the next stop codon would be. And so it turns out to be quite a reasonable challenge to sort of map out the potential consequences. And then you just have to make the argument, which we did, that there's sufficient coverage of all those outcomes in the safety studies that you do to show that that there's nothing untoward going to happen. And so we ended up looking at 3 different patient donors and a full patient dose, which is about 10 billion cells, spread over a sufficiently large number of NSG mice, of course.
Have you used this technology for epigenetic applications, like targeted methylation or demethylating areas? I mean, that probably have a lot of oncology.
Yeah, so we certainly have. I didn't talk about that today, but obviously it's possible to use domains that will recruit and or have intrinsic to them activities that would be chromatin modifying in some way or other. We've done experiments on both histone methyltransferases as well as DNA methyltransferases. So it is possible to target those activities to a location specified by the zinc finger and bring about an epigenetic change. If you follow things like the crab domain, even the crab domain should be able to do something similar.
One more quick.
Yeah.
So one of the points that you mentioned, I assume a lot of people here are interested in, especially getting into genome editing and transcription and regulation. What about the different technologies? I mean, with respect to zinc fingers, TALs, Cas, CRISPR, with respect to specificity, efficiency, toxicity, dealing with heterochromatin in vivo. I mean, what's some of your experience?
Yeah, so I'm slightly biased, right? Just a little bit. The short answer is that all these technologies offer, I think, CRISPR is probably the most uncharted at this point, so it's difficult to know exactly how that's going to play out, although it looks very exciting at this stage. We have done quite a bit of work on TALs. There are these reports that TALs, the TAL DNA binding domain is very sensitive to CpG methylation. We've not explored that in any detail, but I know that others have. I can tell you from the zinc finger perspective that that's not a problem. That the zinc fingers can be made to be CAG methylation sensitive, but by default they're not, and so they will ignore those sequences. And like all these technologies, it's necessary to be. I mean, everything can be applied in to use Martin's first. A lot of it's about dosage, right? So if you can make any of these technologies look good or bad depending on exactly how you deliver it, exactly what dose you want to use, and so I think it's in our hands at least we're very happy with what we've been able to do with zinc fingers, but we also have a lot of experience doing that. So.
I want to ask about timing in the human development when you introduce a change. So, for example, with HIV, you showed within a matter of just several months. You could totally eliminate the HIV RNA from the mouse model, I think it was.
Yep.
But when you're looking at something like introducing exist to eliminate the problems with trisomy 21, do you anticipate that being an embryonic change, or how do you?
Right. So I think we're at this stage where I view that more as a technological tour de force as opposed to a potential therapy at this stage. If I was to dream a little bit about therapy, clearly. One would either have to find a cell type that one felt was both accessible ex vivo, that would be, if corrected, would have substantial impact on the course of disease, or you would have to intervene very early, which obviously is kind of scary to do, but yeah.
Okay, let's thank